Okay, so today we should be able to get through gene expression. We'll talk about our calcium example, and then we'll talk about protein secretion and potentially get into the digestive system. So I loaded those slides on PolyLearn for the digestive system. I also loaded a study guide for the upcoming quiz and something else that will become obvious when you look up there that I can't remember now. So the last time we were talking about this and we said we're going to focus on eukaryotic gene expression, and we said, for example, in eukaryotic gene expression, a gene will often have a TATA box just upstream of the transcription start site, and oftentimes TATA binding protein is the first protein to bind to the promoter region. Okay, so let's go through some steps. And if I tell you steps in a process, I don't care if you go, okay, this is step 2, this is step 3. I'm looking at the sequence. That's all. So as we go through this, keep this in mind. First step we're going to say is the initial transcription factor, oftentimes TATA binding protein, binds to a specific sequence. In the promoter region. And we're saying, for example, this is a promoter region. All right, remember the promoter is a sequence of DNA that regulates the expression of a gene. Okay, so second step. Other proteins can be recruited to the promoter region and bind DNA or bind other proteins. Okay. A general transcription complex is formed. A general transcription complex is formed that contains approximately 25 to 50 proteins. It contains approximately 25 to 50 proteins.
Did you say 30 or 35?
25 to 50.
Oh, 25.
Those proteins recruit RNA polymerase II. Those proteins recruit RNA polymerase II. All right, and then transcription can begin. So I'm going to skip from here. Oh, we've seen this. This is high end, right? That I made. Right, all these transcription factors are starting to assemble on the promoter, and then here comes RNA polymerase II. It's recruited to the transcription start site, and RNA polymerase II makes RNA. So this is why I called it a general transcription complex, right? And these proteins, and you can see some of the proteins are looking for DNA sequences, some of them are looking for other proteins. So the common term there is recruitment, right? That means these proteins are just interacting and basically allowing other proteins to kind of join this complex. When you get up to a critical mass, then RNA polymerase can be recruited in. RNA polymerase in eukaryotes doesn't recognize DNA sequences, it recognizes other proteins. That's a big difference between us and prokaryotes. Prokaryotic RNA polymerase recognizes DNA sequences. That's what it's looking for. Doesn't need help from other proteins. So obviously we're more complex in a lot of reasons, for a lot of reasons, and that includes how our genes are expressed. And again, now if we think about this, kind of the more proteins that are involved in this, the more points of regulation that are possible. Okay, so then RNA polymerase 2.
Step 3.
I don't know what step it is. It's the next step. Right, then RNA polymerase 2 starts running through the gene and synthesizing an RNA molecule. We'll talk about details in a second. Right, so this is our transcription start site.
Can you repeat, you said something about how prokaryotes are different than eukaryotes?
Yeah, prokaryotic RNA polymerase recognizes specific DNA sequences. Our polymerase does not. So it needs assistance in finding that. Right? Now, I want to point out to this right now, and then I'll build on this in a second. But see the way this happens. This is a very localized or proximal part of the promoter, right? So these proteins are close to that transcription start site. That's where that general transcription complex is formed. But there are more distal DNA sequences that can also affect transcription initiation, and that's what this is showing, right? That DNA complex, like, you know, when it's wrapped around the histones and all of that. Well, we're kind of bypassing all of that regulation of transcription. It's extremely important, but we don't have enough time to go through it. So what that means, though, is sometimes DNA sequences farther away from that general transcription start site are also affecting transcription, and it occurs through assistance in recruitment. So we'll talk about that with vitamin D in a second. Okay? So that's what that means. This type of interaction between proteins that are also binding DNA, but sometimes it's away from that proximal promoter. So in eukaryotes, it's weird. You might find a sequence of DNA hundreds or sometimes thousands of nucleotides away from transcription start site that's actually affecting transcription regulation. All right, so I'll make that more obvious hopefully as we go. So now let's look at the next step, right? RNA polymerase has made, here's the RNA coding region. In other words, that's what polymerase is reading, goes through here. And then we end up with this full-length RNA molecule that represents the length of the coding region. So if we look at this, RNA polymerase starts here, it's going to read all through this until it gets to a transcription stop site. And that's in the genetic code. Remember, we saw that before. So it's going to read through. There's a few of them. It's basically a stop codon. And then RNA polymerase is going to go, okay, that's the end of the line. And then the thing disassembles. Then another RNA polymerase molecule could do the same thing. So it can be done over and over again, depending on the scenario. But the point is, that polymerase reads all these nucleotides. It includes exon and intron sequences. And then once that full-length RNA molecule is made, we can just refer to it as RNA. Some people refer to it as nuclear RNA or other things. But we're going to say that's RNA. And then after that's made, we have splicing. Right? So now we have this RNA molecule. Say this is transcription start site. Here's our +1. RNA polymerase bound here. Runs through that. We end up with a RNA molecule. And then RNA polymerase dissociates. Okay? Then we have splicing. And remember, that's when the introns are removed. So in that case, you could just draw something like this. And that yields what we can call messenger RNA. Right? It's shortened. So in your notes, put splicing is an example of RNA processing. Splicing is an example of RNA processing. Can you think of any others that you learned? How about this? Remember that? We call it the poly A tail, the adenylate cap, something like that. Why is this important? You guys talk about it anywhere else? Half-life. Yeah. When I was going through grad school, one of the classes I took was, oh boy, biological regulation, I think they called it. And a guy that was at the cutting edge of half-life research was one of the instructors of the class, so he'd come right from his lab and walk in there. Okay, what are we doing today? Oh yeah, he got a Howard Hughes Medical Institute research appointment, which meant he never had to write another grant. They funded his career, the rest of his career. They said, we'll give you as much money as you need. So one day I went into his office hours, and the whole top floor of what we called the Life Sciences South building there was, he went from a lab of about 5 or 6 people, he's going to have like 25 people in his lab. They're like, your work is great. We're going to support you. Don't waste time doing grants. Just work on mRNA half-life or decay. And one of the things that he figured out, he was one of the people that figured out, this actually helps that mRNA stay around for different parts of time, periods of time. But overall, it tends to be pretty labile, and you guys will talk about this in 329 about how that mRNA structure is actually recognized by proteins and how that can regulate translation. Hopefully you'll talk about that in iron metabolism. It's fascinating, and a few people have won Nobel Prizes for that. But that's one of them. So RNA processing includes splicing, polyadenylation, and capping. Remember that one? This first nucleotide is called 5' capping. It's modified so that the ribosomal structure can say that's start site. So when I say include RNA processing events, you're going to say splicing, polyadenylation, 5' capping. Now, ready for translation. See why it's shorter? Because introns have been removed. Guess who does that work? Proteins. There's proteins that form complexes, and you don't have to remember this. They're called spliceosomes. The proteins are going along going, oh, we know that's an intron-exon interface. There's one end, there's the other end, and they'll pull that intron out, and then other proteins put it back together. And then you end up with an mRNA that is shorter and ready for translation. So then we're going to say translation. Where does all this occur inside the cell? It's all in the nucleus. Nuclear events. Yes?
What is the purpose of the 5' cap?
Half-life. How long the RNA stays around. That's one of them. One of the functions. Yes?
Do those have like a certain order, like the splicing and the polyadenylation and capping, or?
They might, but they're almost all occurring at the same time. Yeah. Yep. Cells are pretty smart. They can do a lot of things at the same time. So some proteins will be bound on the RNA doing certain things while others are, yep. Good there. Right, mRNA is messenger RNA. It's the messenger for translation sequence. And here's translation. It's very simply shown. Okay, so now that mRNA molecule, they're calling it a mature. I would say that's not right. That's basically RNA transcription, not mRNA transcription. Then it's processed to a mature RNA molecule, also known as mRNA, and then that can be exported. It goes out some of those nuclear pores, and again, proteins are trafficking it out of the nucleus, and then it goes into the cytosol, and then proteins recognize that, as an mRNA that's ready for translation, and ribosome assembly starts. So you can barely see the ribosome here. Remember, it's got small subunit, large subunit, and all that. So what happens first in translation? Yeah, small subunit. So translation starts with a small ribosome subunit. Finds the initiating codon. So the small subunit finds the first codon. Then what happens? Then the first tRNA comes in. First tRNA joins, and then large subunit. And then translation starts. Small subunit, first tRNA, large subunit. Now we've got that full structure of the ribosome. Have you ever looked at the structure of ribosome, three-dimensional? They're fascinating. What are ribosomes made out of? RNA and proteins, right? So they're these complex three-dimensional structures that basically allow mRNA to go through, but remember, it's got to have openings in the top, essentially. What has to enter into those openings? The tRNAs have to come in, and then the tRNAs come in. Somebody have a question before I keep going? So now, once the ribosome is completed, the mRNA starts moving through the ribosome. And the appropriate tRNA is matched with the mRNA. And this is all stuff that you probably already know. The appropriate tRNA is matched with the mRNA through the anticodon-codon complementary base pairing. Remember that? So the codon is in the mRNA. The anticodon is part of the tRNA. What's tRNA made out of? RNA and? Close. An amino acid. Each tRNA has a single amino acid. It's loaded with that. It's activated. And so what happens is you've got a nucleotide interaction between two RNA molecules that have nucleotides that can base pair. So it's complementary base pairing. And what kind of bonds are involved there? Hydrogen bonding. That's how this tRNA basically can stay there while it unloads its amino acid. Yep.
Just as a reminder, is the mRNA was copied off of the antisense strand and is a copy of the sense strand?
Yep.
So then
Has sense sequence.
the tRNA is kind of a copy of the antisense? Or similar?
Oh, so this is, yeah, so this is the opposite. It's complementary here. But when you look at the genetic code, this is the sequence you're looking at. You're looking at the sequence of the mRNA. So it's essentially the sense sequence. And the tRNA, so let's say this is, I don't remember the code off the top of my head, but let's say this is GCG and that encodes for glutamate. This would be the one, it would be GCG here. This would be CGC, the opposite, and that one has the glutamate. This tRNA, for example, has that glutamate. So there's more than one tRNA that has the same amino acid. So when you look at that genetic code and it shows, oh wow, there's several different codons that all code for the same amino acid. That means there's like 4 different tRNAs that all are loaded with the same amino acid.
I'm getting kind of confused on like what's like making up the ribosome. So is that like the small subunit with the tRNA?
The tRNAs aren't really part of the ribosome. So let's say, for example, we had a ribosome here. We have the small subunit, and then we have the large subunit that interacts with that. And then your tRNAs are like this. The tRNAs are coming in through kind of openings in the top of that large subunit. And the tRNAs come in. So in other words, you can have a ribosome that's like this with no tRNAs. It's still a ribosome. So the translation complex is mRNA threading through a ribosome, and then translation is when these tRNAs are coming down and matching.
So, and then the tRNA has the anticodon.
Yes, in this sequence. So it's complementary base pairing. Complementary sequence to the mRNA sequence. And then if a tRNA comes in and it doesn't match, it's exported. It's crazy how fast it can do that. It runs through that. And I don't know the physics behind that, about how that can actually happen that quickly. But now look what happens, right? Remember this from whatever. Now let's say we have a ribosome here, and here's our mRNA. And then let's say we have two tRNAs. Remember there's 3 sites. We don't remember all that, the P site, the A site, and all that. But now what happens is, for a split second, there's 2 tRNAs that are right next to each other, and their amino acids are right next to each other. And they're linked together. That's why one of the reasons ribosomes have to have proteins, that's an enzyme that makes that. What kind of bond is that? That's a peptide bond. There's an enzyme that will basically form that peptide bond. That's a covalent bond. And now what happens is, now you've got an amino acid that's, a tRNA, sorry, that's no longer charged, no longer has an amino acid. Ribosome kicks it out. Then it shifts. And it goes to the next spot, same thing happens. Fills that, peptide bond's formed, shifts, empty tRNA comes out. That little video on PolyLearn shows all that and how fast it is. Okay? Good so far? Where's this occurring in the cell? Cytosol. So far, starts in the cytosol. Translation in this case is starting in the cytosol. Good? What's the point of this? You can look at this as just a different way of looking at it, I think. And again, I think that that's not, this is not mRNA. It's made, then it's processed. This is, I think you guys already know this, right? We talked about this before. One nucleotide may or may not change the amino acid, which may or may not affect a protein. All right, so that's basic gene expression and initiation of translation. Okay? Now, first point of this is important. Nutrients can affect, it's got a typo. Trying to make it a point, I got a typo. What should this say? Should be an A. It's affect, right? Sorry. Can affect a gene's expression, increase or decrease the protein being made through altering the basically the efficiency or rate of transcription. We'll talk about that in a second. For example, vitamin D increases the expression of a protein that is needed for the absorption of calcium. Let's talk about this. So let's do some defining our alphabet soup here. See VDR, so in your notes put VDR. Vitamin D receptor. D3. Ready for this? You guys know this already. You had 210. That equals cholecalciferol. There's no way you'd know that at this point. Yeah, so that's a formal term for this molecule. Have you heard of this one? Have you ever heard there's a difference between D2 and D3? Ever seen this? This is called ergocalciferol. Right? Does that make sense? Right now that means nothing. Well, all that means is there's two forms of vitamin D. Their structure is slightly different. Okay? This form, D3, is essentially synthesized in animals. This, plant-based. Do they both work as vitamin D? Yes. And they used to say, probably 10 or 15 years ago, yeah, you know what, we're going to put this in supplements because it works really well. Now they realize D3 is the most effective form. So now you might see commercials or wherever, you know, if Google or whoever is spying on you and you're looking at supplements and you go over and over again and you're going, why am I getting all these things that says, this has vitamin D3 in it? D3 or D, what does that mean? That means that now they realize D3 is a better form, so now they put this in supplements. And we'll talk about supplements later on. But what this means is this is the most active form of vitamin D. That's what that means. And cholecalciferol is made, it starts in our skin. So when we can actually convert cholesterol to cholecalciferol. And then that cholecalciferol becomes active and regulates gene expression. Okay? Sure. D3 is the form made from cholesterol in a process that starts in the skin.
Is that why, like the sun?
Yeah, so the sun is involved in changing the ring structure that gives us a form of D3. Okay? Can we also get this in food? Yep. Good source of vitamin D3 in the diet? Oily fish. What else? Fortified milk. What else? Fortified cereals. Yeah, good. Those are probably the three most important dietary sources for us. Other than that, it's hard to get adequate amounts of vitamin D in the diet. I'll come back to that in a second. Remind me that I need to talk about whether or not we need vitamin D in the diet. Yep.
Since vitamin D and calcium work pretty closely together, is that why it's fortified with milk? Are they absorbed easier together?
Yeah, well, that's what we're going to talk about. How this vitamin D is involved in calcium metabolism.
So
Yep.
if you have like orange juice that's fortified with calcium and with vitamin D, does that work the same or it's not the same because they're both fortified versus the calcium's naturally in the milk?
Effectively the same. Yeah, good question. End result, about the same. Okay, so here's how it works. D3 from the skin or from our diet enters into the blood, goes through the liver, and becomes, we're going to call it this. Calcidiol. What does that mean, OL? That's an alcohol, which means that's a hydroxyl group. This form of vitamin D, as this cholecalciferol goes through the liver, there's an enzyme in the liver that adds another hydroxyl group. So we could do this. It's already got one. We don't see the structure here. Second one is added. This is calcidiol. It's also known as 25 dihydroxy vitamin D, in this case, 3. And you'll see my point of all this in a second. I know this isn't 329, but I don't want to let Lefrano have all the fun. Now what happened?
Oh, an extra.
Diol, 2 hydroxyls. Now there's 3. This happens in the kidney. I don't know why I did that. So here's my point. Vitamin D3 can come in from the diet, or we can synthesize some, but it has to be modified to become highly active. And if you think about why that's important, let's say we go to Avila in the summer and we're out there all day and we synthesize a lot of vitamin D. Can that become toxic, right? Vitamin D is a water soluble or fat soluble? It's fat soluble, and a lot of times I think we know, hey, you know what, fat soluble vitamins can become toxic. They can accumulate to cause harmful effects. How do we protect ourselves from that if we make vitamin D when we're out in the sun? Well, our body has a defense mechanism that you'll learn later on that basically says, this is regulated. So when we're out in the sun all day, it doesn't lead to vitamin D toxicity. What if we take way too much in the diet? We also can protect ourselves from that. But to be honest, this system can be overwhelmed. So vitamin D from supplements can be toxic. Now, with that said, we have a point of regulation here. So when you go to Costco and you buy a vitamin D supplement and it says, hey, it's vitamin D3, does that mean it's this one, or this one, or this one? You'd say, oh, it's the most active. Hey, more bang for your buck. But you've bypassed the regulation. So when you buy it, it's this one. Goes through your gut, into the blood, and then this is not highly regulated. So most of it will become calcidiol. And then the next one, this is regulated. The more calcidiol you have, the more this down-regulates. Okay? And hopefully you'll go into all of that more in 329. If you have questions about any of that, if it's a burning question in your mind, you want to talk about it before next quarter, let me know. We'll go through more of it.
Is cholecalciferol, is that one also the one that's in fortified? That form?
Most of the time it is now. But sometimes you will see some foods that have this. Have you ever heard mushrooms are a good source of vitamin D? And they'll actually take mushrooms and they'll expose them to UV light to convert, basically to go from the ergo form to an active form of vitamin D. So that's common. And that's this one. Does it still function as vitamin D? Yeah, it's still a good source of vitamin D, just not as good as this one. But it still helps. So you can get vitamin D from plants as well. Uh-huh.
My doctor said that like the calcium and vitamin D that I put like from the milk that I put in my coffee doesn't really count because the caffeine like blocks absorption or something.
So what they could be driving at is caffeine can be a diuretic, and maybe that's what they're driving at. I haven't seen clinical data that shows that you'd have to do that. Now, I don't know how much, depending on how much milk a person puts in their coffee, how significant a source that would be. I don't know. Yeah, my mother-in-law, it was significant. Me, not so much. And she'll laugh if she knew I said that. So. Okay, so now let's go through all this. Now, that's the background. Now, the reason I'm telling you that is because this form right here that we're talking about, we're going to say this is the most active form of vitamin D. So we're assuming right here, this has already gone through the activation steps. So even though it says D3, think of that as calcitriol. And here's why. Because now let's say this has gone through that process where it's fully active, it's in the blood, it can now enter into cells. When this form of vitamin D enters into cells, we can look at it like, see, this is the nuclear membrane. This, we're going to say, is the plasma membrane. So you ready? Let's go through our notes. Let's go through our notes, and you're going to say, vitamin D passes through the plasma membrane and into the nucleus. Where it binds, VDR. So vitamin D and VDR combine in the nucleus.
Can you go back, sorry?
Vitamin D and VDR combine in the nucleus. Right? It's vitamin D receptor. That's its ligand. That's what it's looking for. And I can show you 3Ds of this if you want. But essentially, when that receptor finds its ligand, what happens to the receptor structure? See the way it looks like this? And now it's changed. Now this part of the protein shifts. Okay? Next step. The VDR vitamin D complex binds RXR. The vitamin D VDR complex binds RXR. That stands for retinoid X receptor. When you hear the term retinoid, what do you think? Eyes, vitamin A. That's a generic term for some of the molecules that have vitamin A activity. So is there a relationship between vitamin D metabolism and vitamin A? Absolutely. Again, you'll talk about this later, but one of the toxic effects of vitamin A toxicity, softening of the bones, because it alters vitamin D's activity. It overwhelms the normally regulated system, compromises vitamin D's ability to help add calcium to bone, and the person has softening of bones. Okay, so there's lots of interactions between nutrients. In this case, the interaction is between nuclear receptors. VDR is a nuclear receptor. Have you ever heard that term? It's called a nuclear receptor because it functions in the nucleus. A lot of times when you hear receptors, remember last time when we were over there, we said this is the insulin receptor, and we put in the plasma membrane. That's I think the most common time we talk about receptors. Oh, it's on the outside of the cell, binds ligand, signal transduction, whatever. There's a whole bunch of proteins that function in the nucleus as nuclear receptors. That's their side of business.
So does vitamin A toxicity cause bone softening because it outcompetes vitamin D for RXR?
That's
Or why does it cause bone softening?
I didn't go into that, but it alters the stoichiometry between RXR binding VDR and RXR binding its other ligands. This one's called, so we can kind of, you don't have to remember this, but a long time ago I saw a University of Arizona graduate actually is the one discovered and cloned and characterized the RXR. And when they first characterized it, they called it an orphan receptor. They called it RXR because they knew it bound some type of retinoid, but they didn't know which one. Later they figured out, oh, it's a 9-cis retinoic acid form as its high affinity ligand. They didn't know that initially, that's why they say it's X. Retinoid whatever. Okay? So they weren't initially sure on how that happened. But RXR, again, you don't have to remember this, binds VDR, but also binds lots of other nuclear receptors. It tends to form heterodimers. So it'll bind our AR, VDR, and some other nuclear receptors. So if you have too much of it, it'll actually change its presence and ability to bind VDR. Okay, so next step is the RXR VDR complex is phosphorylated. The complex can now bind VDRE. All right, we know an element is a short segment of DNA or nucleotides, we said. Vitamin D responsive means it's involved in being regulated by vitamin D. So here's its consensus or typical sequence right here, these nucleotides. Where are these? In DNA.
What did you say the element was?
See those nucleotides? That's what that complex is looking for. So this is a complex that recognizes specific DNA sequences. And the sequence that it binds, we call the vitamin D responsive element. It's right here, yeah. Okay? We're going to put all this together in 2 seconds. Uh-huh.
Is the nuclear membrane similar to the plasma membrane?
The lipid bilayer. Yeah, it has some commonality, a lot of phospholipids, and its composition would be different, its structure different. The nuclear membrane is a double membrane. Lipid bilayer. And then pores that go through both of those lipid bilayers to make openings. Yeah. Okay, so here we go. One of the questions is, so a long time ago they figured this out. And I again, this guy was at a University of Arizona as well. His name is Mark Hausler. And the first time I saw him, I went, yeah, he's not a scientist, man. He's a Hollywood kind of guy. He walks into this thing, he's like struts in there. I'm like, this guy's really cocky, but he figured this out. I probably walked the same way. So he's in there and he's going, okay, so what we did was we found this out. He's a biochemist, molecular biologist, not a nutritionist. A lot of these findings were from people outside of nutrition. His lab figured out how genes are regulated by vitamin D. And here's how they did it. They basically took cells and they were looking at immune type situation. Remember, vitamin D is really important for the immune system. And what they found was if they took, let's say we had 2 different cell cultures, and let's say both of these are, let's say they're intestinal cells, and we have them in plates and we're growing them. And intestinal cells, if you grow them right, they'll grow in flat monolayers and they'll form actually an absorptive surface. So let's say we have one dish here and the other dish we have here. Okay? Now let's say, for example, we have a mechanism of monitoring and measuring the expression of a specific gene. Okay? And we go, okay, we know how much RNA is being made under these conditions, let's say per minute. Okay? We're making, let's say, 10 mRNA per minute from this particular gene. So we've got a transcription rate. And then they added vitamin D, its most active form. They said, okay, when we added calcitriol, we went from 10 mRNA a minute to 100. Then they did their promoter stuff where they went, okay, we know transcription start site, and they started backing up going, where's the location of this part of DNA that's actually regulating this? And then they figured out, they honed in on it and went, it's this sequence right here. This is what's responding to vitamin D. It's a relatively short nucleotide sequence. We're going to call this the vitamin D responsive element. This is a long time ago. This is, you guys were not even a twinkle in your parents' eyes at this point, right? So they figured this out, and then they went, oh. And then they started figuring out, wow, is this the only gene that has that? What do you think? Would every gene have this? This is located in the promoter region, so it doesn't show it here, but let's say, for example, we looked in greater detail, we looked at the nucleotide sequence. What if we saw it right here? What do we know about this gene? Its expression is regulated by vitamin D. Would you expect to find this VDRE in the promoter of all genes? Are all genes regulated by vitamin D? No. But a lot of them are. So that group is the one that first started figuring out how many genes there are that are regulated by vitamin D. Now we know there's probably a few hundred. So once they started figuring all this, they went, well, we know vitamin D is important for calcium metabolism. Why is it in front of this gene? This gene has nothing to do with calcium. And then they went, oh, immune response. And they started figuring out all the different roles of vitamin D. So if you're doing work and you find that and you work with a specific gene that's got a VDRE, it's regulated by vitamin D. In other words, when vitamin D levels go up in the blood, it alters gene expression. When they put vitamin D and they increased it in those plates and they found alterations in gene expression, that's basically what they were showing. Now, every time I consume milk, if I consume milk on a consistent basis, is it altering gene expression? Absolutely. Mechanism is what we just went through. So here's how it works, though. The last step of this is when the VDR complex binds the VDRE, it affects RNA polymerase recruitment. When the VDR complex binds the VDRE, it affects RNA polymerase recruitment. So let's say this is our TATA. Let's say here's all the other proteins that are involved in this. Now let's see down here, we have VDRE. So with everything that we've learned so far, let's say this gene makes a protein that's involved in calcium. Let's say, for example, we make an RNA molecule, and then we make a mRNA molecule, and then we make a protein that binds calcium. Let's call that protein calbindin, because they did. They went, they followed this thing, and they went, this protein right here binds calcium, it's regulated by vitamin D. Let's say vitamin D isn't present. Do you think that this could still be expressed? So we would call this the calbindin gene. Remember I said there's a general transcription factor complex? General transcription complex. Even without VDR, you'll still get some expression. All these other proteins are general. They're not regulated. Have you ever heard the term constitutive expression? That means constant or baseline expression. Basal expression. Have you ever heard that? And it's like this. Basal expression of a gene. Not this. I know we're nutrition. Not that. That'd be better, right? But this is baseline expression. In other words, let's say we go back to our example and I say, Without a high amount of vitamin D, we get 10 mRNA a minute. Now we add vitamin D, we get 100. Every time you make more mRNA, you're more likely to have more protein in general. So our premise in this case is, well, we said VDR affects transcription. In our scenario, we're going to say in this case, if we have here's our complex, here's VDR, it's a nuclear receptor, it binds DNA, its binding can interact with other proteins that in this case improve efficiency of transcription. In other words, now we have more proteins that are available to help recruit RNA polymerase. So gene expression goes up. So in this case, VDR increases expression of calbindin. Yep.
Oh, sorry. Why do we want to increase the gene expression of calbindin though?
I'll show you.
Okay.
Yeah, good question. So far, so good? Now, what they found in some cases is vitamin D actually decreases the expression of some genes. But in this case, it goes up. So nuclear receptors can cause gene expression to go up or down. And in some cases, I don't know if you've ever heard of this, but a long time ago, a guy actually figured out, wow, you know what? If you have some sun exposure, it can decrease the risk of skin cancer. Kind of weird, huh? And he went through the data, and at first, when he first started saying that, he was at Harvard, and they said, stop saying that. People are going to go out, sit in the sun, they're going to get increased cancer. He said, no, it's true. And they said, stop saying that. He said, I won't. And they said, go. And they kicked him out. So he went to Boston College School of Medicine, I think, and kept saying it. He's right. Some sun exposure is actually protective against certain types of cancer. Not too much. And why? Vitamin D is protective against cancer. And we're not going into that here, but that's... So in some genes, vitamin D decreases expression. In calbindin, it makes it go up.
So this is happening all before transcription?
Yeah, this. Yeah, so we're at the trying to form a transcription complex that's going to recruit RNA polymerase that then is going to start transcription. Exactly.
So the VDR complex is like for more efficient transcription.
Yeah. It's a regulator of transcription. So in that case, it's regulated, it's not constitutive. Yeah. So this regulates gene expression. So at the first part of the quarter, I said, we're going to talk about how nutrients regulate gene expression. This is how. And when I was an undergrad here, they first started talking about, molecular biology meets nutrition. I went, I don't know what you're talking about. And I went to grad school and I went, oh, now we know how specific things on our plate affect gene expression. And now we're kind of at the point where we're like, okay, we understand a lot of this. Now let's look at the sequence of everybody's genes. And that's nutrigenomics, right? You heard that term? We're looking at how nutrients affect gene expression in people with different DNA sequences. That's nutrigenomics, basically the interaction between nutrients and our DNA. Uh-huh.
So then with D2, is it like a totally different process, or does it like just take longer to get converted into like the calcium
Its
channel?
metabolism and half-lives and affinities for specific ligands is slightly different than D3. Very small difference in the structure changes all of those things a little bit. But it does do this. Yeah, just not as effectively. So here's where all this is where we're at. Now let's go to the final step here. This protein, where would you expect to find it in the body? That's a good guess. There is a form there. There's a form in a few different places. That's a brush border membrane. This is an intestinal cell. Nucleus. So I'm a horrible artist. I do the best I can. So I apologize. If I write something you're not sure what I mean, that's understandable. Okay? Oops, like this guy. It shouldn't be here. Okay, so now let's talk about this, right? Most of our nutrients are absorbed in the small intestine. And there's a specific cell type there called enterocytes. And those are gut cells that are absorptive. And so we're going to say this is an intestinal cell. And so this is the lumen of the small intestine. Right? That's the inside of the small intestine. So whatever we consumed, in this case we're going to say calcium in our milk is in the lumen of the small intestine. It's got to be absorbed. It is a charged particle, so it needs a transporter. So calcium actually has these proteins. I'm going to draw right here, that are integral membrane proteins that allow calcium to go through that plasma membrane. Then calcium goes into the cytosol of that intestinal cell and binds calbindin. Calbindin shuttles calcium through the enterocyte cytosol. Calbindin shuttles calcium through the enterocyte cytosol to enhance its absorption. Calbindin shuttles calcium through the intestinal cell cytosol from the brush border membrane. Remember this membrane? To the basolateral membrane. The basolateral membrane, sometimes referred to as the serosal membrane. What's on the other side of this? The blood. So then it goes like this. And again, there's a transporter here, so the calcium can go through here. Now it's in the blood. So the last sentence you probably have is calbindin, transports or shuttles calcium from the brush border membrane to the basolateral membrane and into the blood. Therefore, the more calbindin in a cell, where are we here? Calbindin moves calcium through the cytosol from the brush border membrane to the basolateral membrane, and then calcium enters the blood. So, the more calbindin in the cell, the higher the efficiency of calcium absorption. Yes?
So it just helps it move from the lumen to the blood.
Yeah, exactly. Helps it go through here. So here's the problem. I don't know if you know this, but in general, the calcium absorption efficiency, in other words, the amount of calcium that we eat and how much actually ends up in the blood is relatively low. It's something like 15 or 20%. If a person has inadequate vitamin D status, it goes down. If a person supplements and improves their vitamin D status, it can double efficiency. But we need vitamin D for that. So the vitamin D, one of the important mechanisms, the vitamin D that you consume. So now, if you think about milk, what has to happen? And I'm not going to put it on here, but let's just think about it. So now in that glass of milk, I have calcium and I also have vitamin D, because that vitamin D, that milk, can be fortified with it. So the calcium comes in, it's in the gut, right? The vitamin D is also going to be absorbed. But what has to happen to the vitamin D before it can alter calbindin expression?
Has to go into that.
It's got to be activated. So it's interesting that vitamin D in that glass of milk, let's say tonight, that I have, is not going to help the calcium in that glass. It's going to help it from the next glass. Right? Because the vitamin D goes in, it's got to go into the blood, it's got to be transported to the liver, it's got to be hydroxylated, then it goes to the kidney, then it's activated, then it's got to go back to the intestinal cell. So there's a little bit of a delay. So in general, you're improving your overall vitamin D status when you're consuming that consistently, and you're also, now you're improving your efficiency of that calcium in the milk as well. So those are some of the reasons why they put them together, and that's how it works. Okay. You'll see on the study guide, I expect you to be able to explain all of that. All right, so you want to be able to explain the expression of a gene and how it's regulated by a specific nutrient to increase the absorption of a second nutrient. So it's the vitamin D and the calcium, and you're going to provide a diagram. So if you haven't started practicing this yet, I would start practicing this. Draw out your diagrams and do this. And I might give you half a page or a page to do this, but it's detail oriented, and it's you got to tell me the sequence of events. When you write something on your exam like this or your quiz, and you say, okay, calbindin, the first time you write it, you can write it out. The next time you can abbreviate. But don't just put CB. Don't just put VDR. You got to define it. If you want to put a little legend there. And then off to the side, you can go VDR equals vitamin D receptor, that kind of stuff. Okay, so we always want to spell out abbreviations at some point. That way I know, okay, you're in the club, you get VDRE. All right, any questions on that? Yep.
So what happens when like iron is mixed into this? Does that decrease calbindin, or does that just decrease levels of calcium in the intestine?
So the iron calcium overlap is slightly different. There's a transporter on that brush border membrane called the divalent cation transporter that can transport both iron, inorganic iron in its +2 form, and calcium. There's competition there. It's a good question. One of the things a person probably doesn't want to do is take your iron supplement at the same time you take a calcium supplement. If you notice on those supplements, there's a lot more calcium than there is iron. And so sometimes it can inhibit iron absorption. Yeah, so one of the questions you'll get a lot of, you don't already, is should I take my supplements in the morning or at night? Should I take them with a meal or without a meal? Yes, you should probably alter it. Because some nutrients are better absorbed with an empty gut. Some of them are better with food. And then, so I would say people probably want to kind of move that around a little bit. And I wouldn't take, if a person takes calcium, I wouldn't take all of it in one shot. I would probably do it maybe twice a day in a few hundred milligrams. Yeah.
Can you re-explain the step after the VDR complex binds with the VDRE?
Sure. When VDR binds to its DNA sequence, the VDRE, it affects recruitment of RNA polymerase. So let's say, for example, this protein right here, I did this, I didn't put on DNA for a reason. Because let's say, for example, we're here. Let's say this is baseline, and we have this complex. And now let's say, for example, VDR binds. Well, it'll attract a protein here. So think about how many proteins are involved there. And now this guy brings that protein to the party because it knows where to bind. This one could form a bridge. That could form another one. So now your recruitment of other proteins in RNA polymerase becomes more effective.
And then having more protein just makes like the overall transcription more efficient?
It can, yeah. It can help improve the efficiency of RNA polymerase recruitment.
Okay, and then like before you drew in like the ones that were added, like the proteins above the data box.
Yeah,
So like how are those just
general
Various-
transcription factors.
okay?
That's what we call part of our general transcription factor complex. 25 to 50 proteins for eukaryotes tend to be required to transcribe genes.
Did you say that this complex is only an enhancer for gene transcription?
This one is for calbindin, but if for other genes, that VDR, when it binds, you guys heard the term repressors, right? Repressors are proteins that are involved in transcription that basically inhibit transcription. Enhancers are the opposite of that. So in this case, we're saying that VDR for calbindin expression is increasing expression. Good there? Okay, guys, go ahead.
So calbindin is essentially just a protein encoded by mRNA, or like the arrow, like I'm confused with the relationship.
This one?
No, on the bottom, from mRNA to calbindin, like is it just encoded by mRNA and it's a protein?
Yeah, because this is the calbindin gene. So when RNA polymerase runs through that gene and goes through transcription, it makes an RNA that encodes the calbindin.
So it's a protein.
Yeah, and then it becomes, then it's translated, becomes a protein. So here's translation. I should write that, good point. We got translation. Yep, now we have, so in other words, the more transcription, the more RNA, the more protein you get. In that case, the more calcium can be moved. Does this seem like a lot? Okay, so practice going through it, just kind of go through it, and then we'll talk about the quiz in a minute when we come back. Any other questions before we take a break? Okay, let's take a short break and then we'll continue on. We'll do protein secretion next.
So is the calbindin it's just like function just like shuttle the
calcium.
into the bloodstream?
It'll go from one part of the cell to the other because calcium inside cells is always bound to proteins.
Okay.
And that's there's reasons for that, but calcium is a signaling molecule, so the cell doesn't let it loose. So in this case, it's binding calbindin, which is kind of a shuttle protein.